to the Weekly Bioanalysis, a KCAS Bio podcast.
Hello and welcome to the 94th episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LCMS space for around 30 years. I'm here with my co-host Dominic Guarino.
Hello, my name is Dominic Guarino, and I've been in the LBA, biomarker, and cell and gene therapy space for about 30 years.
Dominic and I are members of the growing scientific advisory team at KCAS Bio. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS Bio's services. Today we're here with a special guest, Jason Hanna, Head of Marketing at KCAS Bio, as part of an action-packed main topic discussion. We're thrilled to have you listening to the 94th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're thrilled to have you here, and we are looking forward to a fun episode today. To kick us off for podcast number 94, Dominic is going to go over today's podcast topic a bit before we jump into the news and resources section. Dominic.
Yeah, hey, today we're thrilled to welcome our head of marketing or our director of marketing, Jason Hanna. And I want all those scientists out there to understand I want to tease you a little bit. You better stick around to learn more about all the exciting things that we're doing in conference season here in 2025. We're going to be attending AAPS PharmSci, as well as the European Bioanalysis Forum. Jason will introduce himself. We'll talk a little bit about some of the approaches we take for conferences, and then we'll take a deep dive into what we're presenting and some of the panels and discussions that we'll be doing at AAPS PharmSci, as well as at EBF. And yeah, it should be a jam-packed show, and we're really excited to have Jason. John, let's jump right in. Go ahead and tell us about this really pretty fascinating little discovery, right?
Well, I love the potential of it. Yes, it's this is not from a usual news source at all. It's really I found it on Blue Sky from some engineering paper, but this is China's oyster-inspired bone glue bonds fractures in minutes. A team of Chinese researchers has successfully created a new kind of medical adhesive called Bone 02. Inspired by oysters, the new glue is designed to repair broken bones quickly without the need for metal plates, screws, or big surgeries. So basically they can inject it directly into fracture site to help speed up bone repair. Bone fragments together in 2 to 3 minutes, even in blood-rich areas where most adhesives fail. And according to the researchers, I always love the link to the animal kingdom. It mimics how oysters stick to underwater surfaces, which inspired the idea, since bones, like oysters' environments, are moist and difficult for adhesives. And this all came out of a team at Sir Run Shaw Hospital affiliated with Zhejiang University, and the research team leader is a guy called Lan Changfeng, Associate Chief Orthopedic Surgeon at Sir Run Shaw Hospital. And like I say, it's all really interesting about difficult environment, etc. Obviously, they say it can work in a blood-rich environment, but it's also the fact that it's biodegradable, so there's no follow-up in terms of digging it back out. So it's just a really interesting in terms of what it might mean for people with broken bones and how it could actually make their life a lot easier in terms of trying to fix them.
Yeah. No, John, I think it goes back to just how nature can solve a lot of problems, right? Most of the drugs we have were kind of in some way, shape, or form nature's helped identify them, and this is just another case of that. I'm when I think of researching oysters, I almost want to fall asleep, but there's a lot of people that work on animals, and we're even working on an environmental study for mussels and clams to look at impact on mutations in genes and stuff like that. But this is slightly different in that someone must have creatively looked at that and thought, how is that working and can it be applied? And
Sure.
What a wonderful space, right? I mean, I've never had anything inserted but you hear a lot of horror stories. You hear a lot about how they just, when it comes to pins and screws, and of course it's advanced quite a bit in our lifetime. Not as invasive to put them in. They don't need to be replaced as frequently. They've developed a lot of things that make them a little more resistant, the kind of microbial resistance, and much more effective. But this just sounds like Elmer's glue for skin repair, right? It sounds great.
It's really interesting, having had a couple of family members go through knee surgery and knowing the recuperation time to get back up and running properly. This is sort of amazing to see this when it talks about it's got strong fixative properties, so it prevents that possible complications from misaligned bone fragments, things like that. There's a lot of, with this simple approach, it can actually address a lot of the issues from um, just broken bones, and like I say, I like the fact it's a minimal. It can be a minimal, tear in the skin to get in there. I mean, it's just anything major medical is required. Obviously, there's a healing time associated with it. So the more minimally invasive you can be, then the better chance your patient has of healing of it. So they're basically saying if they think they want to go into clinical trials to confirm the results, and then hopefully it could make a big difference for patients down the line.
Yeah, no, it's got some interesting sort of, I guess, stats. It has over 400 pounds is what the bond is, which is pretty impressive. And then, John, I don't know what megapascals are, but it's got a shearing strength of 0.5 megapascals. So I'm learning something new.
Yeah, it's like resistance to sliding, and again, it's like if you fixed it, you want, obviously that's what I was touching on, the poorly aligned bone fragments. If it's fixed, you want it to stay in place, which obviously it's strong enough to do that, to prevent, like I say, further complications. If something happens to you when you're within your recuperation, it should resist any shift in your bones. So yeah, I just thought, nice, totally different approach for us to talk about. But again, I love sort of seeing how minds think in terms of adapting things that are out there, and they take inspiration from nature or whatever to come up with something really quite novel.
Couldn't agree more. And you kind of touched on it earlier, the thought of avoiding having to put something that's not really natural to the body, a screw or a plate or whatever it is, and then having to go in a second time to remove it. This just has pretty exciting stuff. And, not that I hope to ever have to use it, but man, this could, I don't know if revolutionize, I don't know the space that well, but I'm sure it would really, really be a much better patient experience if you do need a screw.
That's exactly. It could make a lot of people's lives much easier in the future. Let's move on.
Yeah.
So this is again, we touch on this every so often. Don't forget the old drugs, because they may have further applications in the future. And this is common diabetes drug soothes brain inflammation in mice with multiple sclerosis. So basically, it's metformin. Metformin alleviated brain inflammation in the female mouse model of multiple sclerosis, suggesting the long-used medication could be repurposed to combat MS in people with female sex chromosomes who are disproportionately affected by the autoimmune disease. So metformin soothed multiple sclerosis in female but not male mice due to its ability to inhibit a gene called KDM6A, which sits on the X chromosome. If you have two X chromosomes, the body normally silences one of them so that genes found there are not overexpressed. However, some genes like KDM6A somehow escape this inactivation and are therefore expressed more in people with XX chromosomes than in those with XY chromosomes. So so basically, talking about multiple sclerosis, women are about 3 times more likely than men to develop MS, a pattern that is also seen across other autoimmune disease like rheumatoid arthritis, lupus, and others. This was read by Rhonda Vauxhall, a neurologist at University of California, Los Angeles, who had previously shown that KDMA6 in circulating T cells is associated with autoimmunity in the same mouse model. And in this, she wanted to focus on immune cells called microglia in the brain. So
Wow.
their approach here was there are 23 or 25 drugs for MS. They all target the immune system, Voskuil said. The holy grail of MS is we don't have treatments to combine with an anti-inflammatory that go to the brain. By targeting microglia, she said, metformin could be that treatment. This is all proof of concept, and as she emphasized, she's got some studies going forward, but it might just be that metformin's a proof of concept, and then it needs something that's more targeted that only goes for KDM6A. But it's like I say, it's indicative of what might be a path to address this in the longer term.
Yeah, it's always interesting to see in the article here, it says 1957 is when this drug was first approved. And here we are, I don't know, 50 plus years later, and or maybe, almost 50
Close to
years.
70 if it's 57.
Yeah. Oh, yeah. There you go. 70 years. My math is off.
It's even older than me. Wow.
Yeah, no doubt. Who knew? But no, the fact that metformin has been, that's the WHO's recommendation for treating diabetes, and boom, here we are 70 plus years later, and somebody's figured out how to treat MS. And that, not only is that a kind of an amazing kind of connection, right, John? You kind of talked about that earlier. It's wonderful to think about how others are looking at researching I'll say well-established drugs and using them for other therapeutics. But more importantly, this gets into like patient-centric testing, right? Or patient-centric treating. And this is, women and men are different, right?
Yep,
And
absolutely.
That's a
Yeah.
big part of what this is saying, is like, we really, that's probably like, maybe, I don't know how much in our lifetime we'll start looking at treating, somewhat based off of, your gender, but certainly with as it evolves, I would think that's going to be the new norm. Why? Not only is it within gender, I think race would play a part too. But as it is today, you look at a label, it says take 2 of these. Doesn't matter who you are.
Yeah.
And this is just really an indication of where future of medicine sort of lies, and that it becomes much more, kind of patient centric, and not all drugs are going to be one size fits all, right? We hear that a lot lately in the industry, and I think this is just another example of like, hey, the disease MS itself is more prone for females, so why would you want to not have something that be more specific towards females? And it's just really, really cool how they discovered this. I mean, I think it's still years away, John.
Oh yeah, absolutely, yeah. Yes, I think that's emphasised in the article too. It is like you mentioned that patient centric. I mean, we know there's other medications that where they've figured out within the population what part of that population will respond to treatment. It's certainly the case with the Vertex cystic fibrosis drugs, where they know it's people with a particular defect are the ones who are going to respond to one drug compared to another. I mean, it's just that understanding of the medical conditions and the pathways that might lead to these issues. It will move to that, help the personalized approach, and hopefully not waste doctors and patients' times by administering drugs that wouldn't be effective to them, because we understand who they are better.
Yeah, a couple cool caveats here. Like, it seems like our Rhonda Voskuil, who was at UCLA when she was working on this, was working in circulating T cells, right? And had shown that those are associated with autoimmunity. And she went ahead and connected the dots with MS, which is really, really again, another good indication of how you can connect dots and how people are thinking, and really, really cool stuff. Yeah.
Let's move on. Unless there's something else you want to say, I'll move on to the next.
No, I do have a question for you.
Yeah, sure.
So one of the things, so I don't know much about metformin, But one of the caveats here is they want to kind of maybe engineer it to be directed towards this KDM6A. Is that, do they just chemically modify it? How would they be able to do that? Maybe you don't know, but like,
I don't know. I mean,
yeah,
I think it might be that metformin. I assume probably it might be it targets KDM6A as well as other things. So
yeah,
if you're going for KDM6A, you might want something that's more specific
I
to
guess,
that target
yeah.
rather than a more general. It might, like they say, it might be in one of many pathways that metformin affects. I think it's a question of, well, what is it with the metformin that targets that? Can you then build a molecule around that? I mean,
Yeah.
it's a typical small molecule. It's a pretty routine one to analyze. I know we had a metformin assay years ago at my previous place. So yeah. I think that's probably where she's coming from with this.
Well,
And
yeah, I'm not trying to put you on the spot with that, but it's
no, it's
not like
fine.
it's a metabolite of it or anything like that. And I suppose there's probably
Well,
some,
actually, to be honest, you don't know. I
yeah,
mean, yeah, I mean, she said that we administer metformin, something's obviously impacted the autoimmune piece of it, but it could, I don't know what the metabolites of metformin are, so we don't know if that's what the active agent is or if it's
mm,
metformin itself. Yeah, I don't know. I just thought it's interesting about that.
yeah, and maybe just one other kind of thought or comment
Mhm.
is like I'm sure there's like in vitro studies that can start to tactically kind of
Sure.
look at
Yeah.
how to
Yeah.
modify the drug, right?
Yeah.
Okay.
Exactly. Yeah.
Chemistry's cool, even though immunology is better.
Yeah. It's like LBA is cool, but mass spec is better.
There we go, right at my jugular. No, all right, John.
I'll,
Got
I'll give
another
you,
really
I'll give you on one and take with the other.
yeah, got another cool kind of news here, and something that's really prevalent in the industry, right? It's about anti-AAV antibodies, which is always
Yeah.
on.
Shall I kick it off again?
Yeah.
Yep. Since this is much more your territory. So the new study suggests patients with anti-AAV antibodies may still receive gene therapy. So obviously we talk a lot about gene therapies in this podcast month to month, and it's the headline is, gene therapies are offering new hope to some patients with rare disease, but not everyone. A third or more of patients are often turned away because they've already been infected with natural versions of the viruses used to shuttle therapeutic genes into cells. So antibodies from those prior viral exposures can render the gene therapy ineffective and potentially unsafe. But a new study from the French Research Institute Genethon and the Swedish company Hansa Biopharma suggest a way to overcome that restriction. Actually, I think it's a combination of findings from two different studies. But the French one was led by Jeremy Docau, a pediatrician at the Anton Becler Hospital in in Paris. My tongue seems to be sticking to the roof of my mouth today. He tested a new approach in a 22-year-old woman who was previously ineligible to participate in a trial of Genethon's gene therapy for her ultra-rare disease, Crigler-Najjar syndrome. That disease is caused by mutations that break an enzyme essential for metabolizing bilirubin. So as you read down it, if you get too high bilirubin, it can actually lead then to, I think, brain damage. So it's if bilirubin levels are left unchecked in people with Crigler-Najjar syndrome, the molecule can cause irreversible brain damage. So one of the things that they do right now is blue light phototherapy that turns bilirubin into a form that is more readily excreted from the body, but the process is time consuming, akin to lying in a tanning bed for half a day.
Oh,
So it's something that can be managed, but obviously if you can get a gene therapy, or sort of cure, it's a better way to go.
yeah. So
So
it's
just, just
it's
sorry, go ahead.
interesting. Yeah, it's interesting because there's been a lot of evolution in AAV therapies, and people have moved away from pre-screening because they've engineered AAVs that there's a known set of kind of anti-AAVs that they just simply knock out those regions within the
Okay.
AAV itself. So
Sure.
within this, it kind of, I read something about this is the first time that somebody with pre-existing antibodies has been treated with AAVs. That may be true, but I think this might be a more natural form of it. So there's been a couple things to overcome because we all kind of have anti-AAVs, every single one of us, and that's why
Sure.
sometimes they use 8 versus 9. And I'm not a super expert in this space, but they've even kind of made hybrid versions to overcome it, right? And so rarely does someone have all the AAVs.
Yeah, sure.
But that they even kind of caveat what they said, that there's no standardized kind of testing of anti-AAVs, so people have different
Mhm.
titer levels. So they kind of talk about that, but it's still like one of the real kind of benefits from this is that it seems like this could be a universal way to overcome it. But like I said, there has been some advancements where people who have they don't. I think a good percentage of the trials don't even look at pre-existing antibodies because they just know they're not neutralizing, and they've been able to overcome it. But that again, that's probably a little bit naive, because if someone's unresponsive, then they want to look at, well, you still
Sure.
got to do
Yes.
immunogenicity for
Yep. Yep.
for therapeutic induced AAVs. It gets really complicated. Not complicated, but it's hard to be able to develop a method that's for pre-existing anti-A. This is like a golden, the holy grail of bioanalysis for supportive AAVs would be if you could design an assay that says, hey, these are pre-existing
Mhm.
AAVs, and then
Yep.
these are therapeutic AAVs. Although one could say, when you screen a patient at baseline and they don't have anti-AAVs and then they're induced, you can indirectly say, or probably pretty directly say, that they are indeed therapeutic induced AAVs. Versus if someone has a titer and at baseline, and then the like the does it increase again, you can indirectly say, hey, those are anti-therapeutic AAVs. But even then, if the drug's still working, those could just be simply more endogenous anti-AAVs, you could argue. So it doesn't have a lot of clarity when it comes to looking at anti-AAVs. But it's an incredibly hot, I would say, hot topic. But like I said, there's been really a movement away from it, because there's been some engineering and even showing that pre-existing AAVs often aren't neutralizing of the AAV. But clearly, I learned something new in this article
So,
here,
so,
that it's
so,
the
yeah. So, do you want me to talk? Let's touch on what they actually did on this.
sure.
So basically, yeah, their gene therapy uses AAV8 to deliver, uh, as the delivery component to, obviously, for basically delivering the therapy. The patient, this is a single patient, so there's really not a huge,
Mm,
not a huge set we're talking. But she was excluded from the original trial because she had antibodies to AAV8. But she was included in the new study because they administered an enzyme called imlifidase,
mm,
which is actually a Hansa. This is where Hansa come into the equation. Hansa Biopharma actually developed imlifidase, and that temporarily cleared out the antibodies, allowed administration of the gene therapy. I think so that then temporarily lowered our bilirubin levels, and I think it meant that I it meant that she could do. Um, I think there was she. It meant it was possible for her to go on honeymoon without having the 12-hour blue light treatment to
mm.
address bilirubin, things like that. So it did, with this approach, it did at least temporarily help with her life, not lifestyle, but life quality.
Yeah.
I think there was it as she as I think as time went on, there seemed to be a diminishment of effect. But again, it's like one patient. At least it's showing a potential. If, like, say, if you're talking different AAVs that get away from pre-existing ones, that you just take that approach, then that's you basically got multiple shots on goal for
Yeah.
helping these
Yeah.
people, because that's really what you're looking for in a case like this. It's like, how can we help these people with these rare conditions? We don't really want to exclude them from a potential treatment. Is there a way around
Yeah.
in their body that we can get that treatment to them. And I think that's really what this is illustrating.
Yeah, and it's an ultra rare disease, Kringler-Najjar syndrome. I'm not quite familiar with that, but it is the mutation is in an enzyme that's essential for metabolizing bilirubin. So that's how it's all kind of tied together. Even though it's still a single patient and in an ultra rare disease, which is a new term for me. I didn't know there was ultra rare versus rare, I guess, but it's still a fascinating story, and just goes to show you how like you said, bringing unmet needs, unmet therapeutic areas, and how to overcome therapies that could be beneficial. It's a wonderful story, and hats off to these two companies.
And just to, there is a little bit more in imlifidase. So it was originally developed by Hansa as a way to clear antibodies. I'm sorry, my
Don't worry about it.
Hansa originally developed imlifidase as a way to clear antibodies prior to kidney transplantation and has a conditional approval for the drug in the EU. It also is working with other companies like AAPS Bio, Genethon, and Sarepta to test the enzyme as a pre-treatment in gene therapy. So, I mean, they're definitely thinking, what can this drug do to help patients? Because, I mean, we've seen there's been quite a few instances with gene therapies recently where there's been obviously talking small patient populations, but there's been some severe adverse effects. So if it can help with that kind of thing to help with
Very
those
true.
treatments, then it's definitely worth investigating.
Yeah, no, I think as AAVs are going to, they will be dominating medicine in the foreseeable future, along with maybe ADCs and other things like that, ARCs. But
Yeah.
they have a big application, and to be able to have something that allows you to ensure that doesn't matter how many pre-existing antibodies you have, the drug will still be effective is a wonderful story and a
Yeah.
nice advancement in
And
science.
and I will stop after this, but there's also another French company working on its own version of imlifidase, dubbed that they're called Vivet Therapeutics or Vivet Therapeutics, and they also shown results of a phase 1 study in healthy volunteers, showing that imlifidase could reduce anti-AAV3B antibodies and provide a window of 2 to 5 days for potential gene therapy administration. So it looks like there's a fair amount of, obviously people looking at this area to see what can they do to help with
Well,
the gene therapy.
yeah, that's interesting. It kind of occurred to AAV3s are rare, and probably because there's a lot of pre-existing antibodies, so people stay away from it. But it opens the door up to use AAV3s, right? It continues to allow for just expansion in the AAV space. It's good stuff, John. But maybe
So,
we got
so
time for
yeah,
one more.
just very quick question. I obviously know nothing about AAVs at all. What's the difference between AAV3 and AAV8, and
Yeah,
what drives the nomenclature of them?
yeah. It's just their spike proteins, right? How they're
Okay,
it's not earth shattering there. It's just they're just slightly different changes in the vectors themselves between you know, like any sort of ISO forms of things. There's
sure.
I don't know about one, and now you're challenging me a little bit, but it has to just do with the like there's like herpes virus 1 and 2, there's AAV virus 1 and
Okay,
2 and
yeah.
3 and stuff like that. And I'm it's a good question, and I think I've answered it right. But if somebody calls me out on it, I'll research it further. But it has to do with how they're presented and the different I call it a spike protein, although an AAV might be slightly different format. But yeah, it's
Okay,
just the
okay.
surface of the viral vector has different proteins on it.
Sure. Okay. Fine. Shall we do one more news item, or do you
Yeah,
want to go?
this is
This,
a really cool one, and one that's near and dear to me.
that's why we thought we'd touch it.
Yeah,
So this is, three scientists win Nobel Prize for discovery of regulatory T cells. The 2025 Nobel Prize in Physiology or Medicine has been awarded to three pioneering immunologists for their work on regulatory T cells, including two who conducted their winning work at Celtech in the UK. So you got Mary Brunkow, PhD, Fred Ramsdell, PhD, were at Celtech, who are now part of UCB, and they pieced together how a gene called AAP3 controls the creation of regulatory T cells, which suppress T cells that inappropriately attack the body in a vital function known as peripheral immune tolerance. What's interesting is that this is an award for really, it's for sort of when they talk about it at the end of the article, it's for discovering a principle. Both Mary Brunkow and Ramsdale have both left Celtech/UCB, and Ramsdale, I'll stop calling him Ramsdale. Fred Ramsdale co-founded Sonoma Bio and previously served as the biotech's chief scientific officer, and they're trying to turn his Nobel-winning work into novel therapeutics with a regulatory T-cell therapy called SBT-77-7101, currently in phase 1 trials for rheumatoid arthritis and hidradenitis suppurativa, along with other Regeneron partner programs in the work. So it's like I said, this is rewarding what they've found is that the principle. And so we haven't shown that it has any therapeutic application yet, but it's obviously early days and it's being looked at. And I thought at that point I'll pass it on to something.
So, it talks about how in 1995 they discovered different T cell populations expressing the IL-2 receptor or CD25. So, John, that's like I started in 94. We were doing autologous T cell therapies. We were seeing and I won't go into great detail about how that therapy worked, but we'd see these T cell populations that popped up that were like, what the heck are these things?
Yeah.
There weren't nearly the amount of tools you have today. And fast forward to 2001, I'm in my second year of my PhD program, and I'm being taught about regulatory T cells. And right around 2004, 2005, I started looking at FOXP3. It's an intracellular cytokine, intracellular protein. So you got to do some intracellular staining by flow cytometry, which was still, it's come a long way in the last 20 plus years. But back then it was a pretty intense, it still is to some degree, a really complex flow method. And even at Streck Labs, I researched FOXP3. We're looking at ways to stabilize it with the Cytochex BCT tube, which it was unable to do. But FOXP3, it's like this is happening in my lifetime, right? It's like, oh my God, yeah. I can't tell if science is moving really fast or I'm just really old, maybe a little bit of both. But it's always fascinating to see how awards for work that dates back, 20, 25 years. And then there's an interesting statement. They said there were a lot of researchers that was skeptical about regulatory T cells. I remember that in my first year of grad school. In fact, I had an immunology professor, a world-renowned professor in the mucin space, who really told me I was wrong. And I'm like, wrong? I got proof that I'm right. And so the young scientist is like, oh my God, am I gonna fail? Right? It made me remember a story where I kind of missed a significant question because she was like, you might be right, but that's not what we taught you here. And I was like, oh God. And that's when I realized academia might not be the path for me. Right. Yeah. But back to this whole, Sonoma Bio's working on it hard. It's just yet another, and I would say that the regulatory T cell pathways are still evolving. That's not fully vetted out. There's still, and then even post-grad school, there's now what they call the T helper 17 pathway. That was when I got my immunology degree, that really didn't even exist. There was maybe a mention of it. So I would be surprised if somebody doesn't win a Nobel Prize for T helper 17, which to me, they're all commingled quite significantly. And what I mean by that is there's think of them as T cell stem cells, right? So there's just progenitor T cells that if you give them certain cues, certain cytokines, they'll become cytotoxic. They can become helper, they can become regulatory, and they can become even this T helper 17. And what's fascinating is if a given cell, whether it's one of those 4 types that I was sub T cell types that I'm talking about, they can actually de-differentiate in the right environment and then go into a different path, meaning a T helper cell, if stimulated with certain cocktails of cytokines, can revert back to more of a progenitor type. And then if you introduce a certain set of what would be more in the T helper 2 cytokine pathway, you can generate a T helper. If you cue them with like TGF beta, they tend to make regulatory. So it's really a fascinating thing. I guess what I'm saying is it's not like linear, it's
Sure.
not stagnant. It's very fluid like, and so just really cool. Love to see immunologists win Nobel Prize, and
But I do have to step in and acknowledge that there was a third
mhm.
prize recipient. It wasn't just Mary Brunkow and Fred Ramsdell. They were also joined by Shimon Sakaguchi, who in 1995 discovered that a population of T cells expressing the interleukin 2 receptor alpha chain, known as CD25, were responsible for protecting mice from autoreactive immune cells. Without these cells, mice became terribly sick with autoimmune disease, their T cells attacking their own bodies. The tolerance promoting cells became known as regulatory T cells or Tregs. So it was a precursor to Brunkow and Ramsdell. This is where you're talking about that skepticism about
Uh-huh.
the existence of regulatory T cells. And then once they got the role of FOXP3 was shown, then Sakaguchi followed up to confirm in 2003 that the gene is necessary for Tregs to develop. So it's like each of them building on each other to come up with a more complete picture, and like you
Yeah.
say, to help move things forward. So
We've been given we're going a little long, John, but I could sit here and talk a lot about what you just said. The 2003, it reminded me how heavy it was researched and how it appeared to be a very promising target for therapeutics, because people would say regulatory T cells suppressed the effect of acute T cells, right? And so that a lot of immuno-oncology patients have high levels of
sure.
regulatory, and they were trying to knock it out. But as I mentioned, it was an intracellular protein and it became a tough target. They found other ways to knock out T regulatory cells. And I think there's a number of drugs on the market, although I'm sure Sonoma's still going at it. But let's make sure we
Yeah,
get
sure.
some time for our
Absolutely, yeah,
guest.
yeah, absolutely. I
Yeah.
wasn't meaning to drag things on, but
No, maybe,
these are all fascinating
maybe it's because
topics. But
maybe it's because we're recording on a different day. We're a little fired up here on a Thursday, right? So,
yeah, yeah.
yeah, well, let's welcome our guest. We're pleased to have Jason Hanna, head of our marketing, director of our marketing, joining us today. We're going to talk about some of the conferences that are going. The conference season is upon us, is how we like to think, for 2025, and we're going to go over all the exciting things that we're going to be attending. Most notable is the AAPS Pharm 360 meeting and the EBF Forum, which are probably two of the biggest, along with WRIB, which is a different time of year. But Jason, do you want to maybe?
Yeah, I think I called it conference season simply because yes, we're at conferences all the way through the year, but November is where it is a huge
Oh yeah,
focus on just because PharmSci 360 or APS almost coincides with the European Bioanalysis Forum
oh
open
yeah. There's a whole
season,
host of them going on, and
and
these are just two. Yeah, for
it's just
sure.
they have a big impact on the lab. That's why I particularly sort of said it's conference season right now. But
Yeah.
anyway, let's at least get started. Jason, as we do with all of our guests, it'd be really good if you could give us a perspective on what your background is prior to joining KCAS Bio, and then what your current role is, and just talk about what you and your team do.
Sounds good, John. Thanks for that, and really appreciate being here with both Dom and John. I've had the opportunity to travel with both of you to these conferences, and haven't yet been on the podcast, but I am a big fan. So it's really fun and good to be here with both of you, sort of virtually, I suppose you could say. Looking really forward to the conversation, and I think we've got a lot of great things going on as the year winds down. It's definitely been a whirlwind of a year in a lot of ways for the industry, but I think there's some good things ahead, and certainly as we kind of zoom in on the rest of the year, the conference season's a lot of fun, and I think will be a nice finish to the year. So I'll just keep going here, John. As you said, thanks for kind of teeing that up for me. My name's Jason Hanna. I'm the director of marketing here at KCAS Bio. I've been here just over 2 years, and again, another whirlwind. It just seems like it's flying by so fast. A lot of fun stuff going on. I was aware of both of you gentlemen before I joined the organization. Definitely did my homework, prior to 2 years ago joining, and stumbled upon the podcast here. So again, been a big fan for a number of years. It's a lot of fun. I've been in a handful of different industries throughout my 25-year career or so in marketing and sales. And most recently, or I guess preceding KCAS Bio, was in the financial tech world. So some regulations, a lot of that similar to the science side of things with the quality, and regulatory, compliance components behind it. Have a lot of that in the financial world as well. As with the small startup, we were a Series A and ended up going through acquisition a lot sooner than we had anticipated, and then shortly thereafter found myself here at KCAS Bio, looking to really apply the sales and marketing techniques and principles that I learned in that industry and other industries to KCAS Bio to help us grow. And I know on this show here you've talked about a little bit of the history of KCAS Bio, here and there. And so I think we have some great things ahead, and I'm just happy to be a part of the rocket ship ride, that we're on.
Great. So, Jason, why don't we kick it off with what are some of the approaches we take for conferences? And certainly I have my thoughts on this as well, and whether it's in the US versus Europe. So how does that all work?
I do, and I think it's interesting because obviously we see a difference in how our customers react in the US versus Europe, and right now we have these two conferences back to back that you can't apply the same approach to both because your customer base have got a different way they assimilate things. So just thought it might be quite nice to touch on how you approach the two different conferences in two different continents.
Yeah, this is a great question, and it's so funny, John, that you mentioned the customer base, and that's where all marketers talk, right? If there happens to be any other sales or marketing folks listening in on the podcast, I might sound a little cliche here when I say, it's customer first, but it really is, right? And so when we approach these conferences, we want to do this from the standpoint of how can we represent ourselves in a manner that's relevant and beneficial to our potential sponsors, and as closely as we can relate to present ourselves to the content or the topic or the audience type that will be at these particular conferences or events. So here's where I might diverge a little bit from your thinking, though, John.
And I have a follow-up question, which I just popped into my head, but keep going. Yeah. I'll jump in with it.
Yeah, well, here's what I was going to say is that the process to go by to whereabouts we go by preparing for conferences is the same, in US, Europe, Australia, wherever we may be. We want to look at it in terms of a marketing campaign, sort of analogous to the scientific method, right? From a marketing standpoint, what conferences make sense from the audience where we can most represent ourselves relative to the services that we offer. And then we look at pre-show planning, during show activation, and post-show follow-up. So there's some very methodical steps that we go through in each of those three phases of conference and event planning. And so that's the same, really, no matter where in the country you may be, right? But where your point is on the nose here is definitely with the audience type, right?
Yep.
So right, and so that maybe is what we can dig into a little bit. I know, John, you've been to the EBF conference in Barcelona year after year, and so I would be curious to kind of put that question back on you and see how that is different than the conferences in the US. But you had a question for me back first?
Yeah, I think I was thinking again difference between the actual two conferences we're talking here. Where APS there's obviously the scientific stream that goes on around the there's a lot of different sessions. There's a very broad scope of topics. But then we're mainly there for like the trade show where you're on a stand. Well, the trade show piece of it is huge, is a huge part of the conference. When it comes to EBF, it's the talks, etc., dominate, and the trade the obviously the booths are important because they the one really nice thing about that is that they schedule activities around the booths, so there's a really good chance for people to interact. But that science is first and is really to the fore. I always found, I've been to APS a number of times. I always found it a little bit of a difficult conference to navigate because I'm there for the trade show, but I never quite know where anything else is happening.
Yeah. Now, John. Now they've made it very bioanalytical these days. So
Yeah, well, that's changed from when I went. You know, where bioanalytical was quite minor in it all. But yes, it's sort of yeah, it was.
No, it's evolved. It's actually because they combined the NBC with it. They've made it a lot more. Or actually, they split the NBC out now, but they've held on to some legacy topics that a good chunk of it is dedicated to
Sure.
APS. And now there are, I don't know, I think there's almost 20 kind of focus groups. One which I lead is the critical reagents, so that that presence, like there's the PK one, there's an ADC one, there's a total and free, there's a NAB and immunogenicity. So all that's kind of updated, and a lot of us go because of that. So it's gotten a little more heavy on the BioA side. But with that being said, there are plenty of pharmaceutical science there, and the alumni of the University of Michigan Pharmaceutical School is not of interest to me, to your point.
Yes, I suppose my question really comes from, it's like, I'm trying to think of this as I'm talking. It's like at EBF, it's like, what can you know the people there really mainly talk the science. What can you do to pull them in to talk to you at the booth? And then similar thing at PharmSci 360. What are people mainly there for? Is it the science? Is it the trade show? And again, what can you do to pull the right people in to talk to you, such that something comes of that conference presence?
Yeah, this is a good question. Here's how I'll try to address this. It all starts with those three phases that I mentioned just a minute ago: the pre-show planning versus the day of, or the during show activation being phase 2, right, and then post-show follow-up being phase 3. So I would say that in this phase 1 approach, we do look at the show holistically, no matter what the format may be. You know, what type of presence can we have overall? And it really starts with a relationship with the association, in this case, the EBF folks or the AAPS folks. And so we have a plan that we build out in conjunction with them as best as we can in advance, of any given show or conference that we go to. So we might, for example, on one hand, want to look at what thought leadership opportunities we have, where we can have our talks. I know both of you've done this, ad nauseum, right? Various talks and presentations that you've done at that, but that is part of marketing's responsibility too, is to see how that fits into the overall show. Like, marketing is not just responsible for the booth, right? Or just the signage, right? It is really the holistic view back to what really matters to the customer, John. Like you said, what do they want to hear? What do they want to learn about? What's new in the industry? Some of the things like you talk about on this podcast. We want to represent ourselves holistically as best we can at these shows and conferences. So we might say, okay, we want to have this literature, at the booth, because it resonates with or aligns with what John or Dom is talking about in one of their talks or a poster presentation or whatever our ops people may be doing with posters and whatnot. So it really is that kind of a holistic approach. But where the divergence, John, to your question, I think is the activation and maybe some of the dials that we turn up or down at the various show types. For instance, EBF is a smaller show, like you said. The kind of the food and beverage area and the break times and all of that sort of meander around in the exhibit sort of area, which is really just like a large conference room, wouldn't you say?
Yeah, absolutely. Yeah. It's not a big space at all.
Yeah, exactly. So it that has a little bit more of like kind of a quaint sort of networking feel to it.
Yep.
And so that's good, and we take advantage of that, and people are in there. They're wanting to decompress from, the very scientific talk that they just heard, or gearing up for the next one that they're going to. Whereas at AAPS, at least the PharmSci event in the fall, typically that we're talking about here, is more of a traditional trade show with a huge exhibit floor. And so it is, it is different by nature. We may not have some, quaint conversations, right, with people that we've known year after year, right? It's going to be maybe more of some new people that we're introduced to. Our approach is going to be a little bit different. Maybe we have different handouts, that we may have, or more just kind of high level. Who is KCAS Bio and how can we help you? Might be something that we prepare for at a larger show like that. So there's a lot of similarities. It's just really a matter of those dials you kind of turn up or down, or where you kind of focus in on your day of activation, depending on the format of the show.
Oh, great! Sorry, I got a little bit distracted. I had another guest in the podcast who has since been kicked out.
I take it that was a cat. No, I'm just kidding.
Black and white cat Alfie was demanding to be part of it all, and after time on my legs and on my shoulders, and he also has this unfortunate habit of nibbling on my legs, which is not great. He's now gone, so I'm now back in the moment. So let's move on. So obviously, like I say, two big conferences for us. First up of the two is AAPS, the PharmSci 360 conference. What do we have happening at AAPS?
Right. Yes, we have. Let me cue up my note here, because these titles are always so long.
Yep. It's really difficult to come up with a succinct title for a bioanalysis talk. Believe me,
everything you wanted to know about ADCs, but we're afraid to ask. That's what you're going to say, Jason. But tell us.
Well, you had actually, was that your title, Dom, at the Scientific Forum AAPS?
One of them was like something like everything you wanted to know about X, Y, or Z, or something like to qualify or validate. That is the question.
That is the question. Yeah. That was. See, I love that. That resonated with me as a marketing person. These kind of catchy titles, but it was also extremely relevant back to what the customer wants to hear. That was a topic that had a lot of good attendance at that particular talk there, Dom, a few months ago. So anyway, coming up at PharmSci here, we have Don Duffield presenting bioanalytical strategies for therapeutic drugs and their conjugates, ADCs, oligos, etc., by QQQ and HRMS strategies and case studies. So I think that's going to be a pretty good one too. She had a great turnout last year in the talk that she did at PharmSci. So looking forward to that one.
And I'll also, there's also the as I mentioned earlier, the community always gets together, so that's something interesting to check out. You can see updates first if you're not familiar with all the great AAPS focus groups that are out there. You can learn more about. Again, I think it's close to 20, maybe it's a little less, but each of us has to go up and give a 5-minute presentation on what we've been up to. So it's a great way to learn about areas that might be of interest to you in bioanalysis, and you can, you're happy to join any of them. But as I mentioned, I co-lead the critical reagents, and we're in the process of working on a manuscript, so you can learn all those great things. It's also for anyone, it's a great way to want to contribute to the community as well as participate in manuscripts, which are always good for one's resume. So that's another thing in terms of what we'll be doing at AAPS. So I do have to give a little 5-minute podium, but and I know I think Dawn and I are still working on if I'm going to add some immunogenicity of ADCs to her presentation, which we've got a lot going on there in terms of just some of the things like domain specificity. And again, I'm always big on critical reagents and some of the tools, and that's an interesting topic to ensure that you're not panicking. And what I mean by that is if you need to go make a critical reagent, that can take anywhere from 3 to 9 months, and that often is going to be very problematic. Anything else with respect to AAPS? Or I know we're running a little short of time, so maybe we could shift to EBF.
Sounds good. Yeah. John, I suspect you'll have the kind of the same question there. We have
Sorry, it's exactly the same question because
Well, good. Well, as you know, you're one of our talks there. So, what's your title?
So EBF's an interesting one because it's like when they're asking for abstracts, they have set they actually have a set of ideas that they have for their sessions. So one of the challenges when that call for abstracts comes out is, do we have anything that can fit in with these session titles that they've already come up with.
Mhm.
And the other nice thing about EBF is they really don't like talks that are salesy, although things do sneak through from year to year. But if you can talk about something that has been a problem for you that you've then turned around and solved, or it shows a path forward, and it's usually a good way to approach a talk for EBF. So I think we I'm trying to think what we anyway, the title that we went with is considering importance of considering environmental factors during LCMS method development biomarkers, and this basically we were very quick background. We were asked to develop an assay um, for some biomarkers in CSF, which came up with a method pretty quickly. But when it came to actually in the matrix, there was major issues with contamination throughout, and it wasn't just in matrix; it was also just in standard solutions, because you're doing biomarkers where it's like parallel between the matrix and then a surrogate matrix. A lot of the issue was with the surrogate matrix, so then there's a lot of troubleshooting associated with that and pinning down where the issue lay. So that's the gist of it. And it sort of, again, it was scrambling to find a session that fit in, but it's in there okay. The conference has changed a bit this year. It used to be everyone turned up on the Tuesday, but Wednesday, Thursday were the big conference days, and then it was a half day Friday. They've actually expanded it to 3 days, so it's now Tuesday, Wednesday, Thursday are the main days of the conference, and then everyone can leave Friday for a long weekend. So my talk is on late on the Tuesday afternoon, which is great because it basically it's a talk on the first day, and then I'm free for the rest of the week to enjoy social activities, which is also key because I'll let you. Well, basically, we have a happy hour. I think, is it on the Tuesday? We're inviting people to come and join us for that. I'll let you talk more about that, Jason, and
Yeah.
some of the other presentations.
Yeah. Well, thanks for all of that lead in. Yeah, you have that particular talk, and then we've got two other KCAS Bio representations, you could say, at EBF. One is a workshop that one of our scientists out of our Lyon, France, is hosting there at EBF. It's on ADA assays. And then thirdly, we have a poster presentation on ultrasensitive assays. So we have quite the coverage there.
Yes, so this is fascinating bioanalysis here, John. You and Jason. So you mentioned, John, your biomarker one is a really cool one. I think I really do think that's something that recently someone had either caught wind of that or had been asking me about that, and so I meaning biomarkers can be challenging because, as you mentioned, the environment and there's just some unknowns in there. And I don't know if you're able to talk more about that, or actually, for the sake of time, we won't. Maybe we'll.
And I won't, because I'm not going to spoil it for the audience.
Yeah, exactly. And then
for the audience, ABF.
Yeah.
But I will say that if you're listening and you're in Barcelona, come and see my talk. I think it's like somewhere after 4:00 on the Tuesday afternoon.
Yeah, and then there's Jason, you mentioned a workshop, right, on was it peptides and ADA, or was it right? That's what it was about, or did I miss that?
ADA assays and therapeutic peptides.
Yeah, no, really challenging stuff there, because the size of an ADA, the size of a peptide, has very small real estate to be able to establish good positive control against it. But as the potency of these peptides continues to evolve, the FDA is asking for ADAs, or anti-drug antibodies, against it. So it is a tricky sort of target or drug therapeutic to look at in terms of developing ADA assays against it. And of course, I'm always going to think about the net evolution. They've actually started, we've got some cases of doing where they're attaching peptides to antibodies. And so there's antibody drug conjugates that include things like payloads and siRNAs, they're called ARCs. And then there's actually peptides that are very incredibly evolving areas as well, and they're challenging bioanalytical. bioanalytically, and we've got a lot of expertise, and no surprise that we're hosting a workshop there. And then last but not least is the NULISA system. And those of you that aren't familiar with the NULISA, this is I call it an ultra sensitive platform, but it's even more than that. And what I mean is, not only can it get down to sub-picogram, sub-femtogram levels, allomer levels, attomolar levels, I guess is what they say. So like that is an incredible amount of sensitivity, which is really good for screening of biomarkers and finding good biomarkers for your therapeutic. But in it, really one of the biggest powers is within a 25 microliter sample, they have some panels of up to 156 or even up to 250 quantifiable biomarkers. So that's unprecedented. There's only one other system on the market that can get that type of readouts, but that's a semi-quantitative system. So this NULISA is incredibly promising. We're one of the first CROs to have it. And not only does it have application for, they've got some pro-inflammatory, that's the 250. They got a neurodegenerative panel. So it's really good advancements in science. We're only as good as the tools that we have. And then in addition to it being really application for biomarkers, they have home brews. And those are, what I mean by a home brew is that they sell you a little kit. It's very similar to what you can do with a Simoa system, for instance, where you get ultra sensitive for your biomarker. But we're going to expand it into some PK analysis. So maybe we got another tool in the tool chest for PK. So really do encourage anybody who's interested in these types of things to be sure to either, not everybody can attend EBF, but certainly reach out, and we'd love to share some of those wonderful tools that we've got. So we've got a few more minutes of time. I want to be cognizant of that. I think we've covered most of our agenda, but what else do we want to do before we put Jason in the hot seat? I think
it's just, is there anything more that you want to say, Jason? Is there any other activities that we're doing that support these conferences out, in parallel with the conferences themselves? Just let us know anything that's going on, and then like I say, we'll move to your torture chamber after that.
I'm ready for that. Well, no, I think the last thing, John, just to answer your question from a minute ago, was the happy hour that we're having. So I won't spill too many details. It is a special happy hour. We don't have the opportunity to do as many of these in Europe, so we're really excited about this one at EBF. And all that I'll say is go to the resources section of kcsbio.com, and there's a registration form there. By the time this podcast episode comes out, there should be still plenty of time, but register, please, and enjoy a really nice happy hour in Barcelona.
And I suppose the other route is if you come by our booth at EBF, then there's also the route to be invited when we're talking to you.
Exactly.
Yeah.
Yes.
So there's mechanisms. Yep.
Exactly. Thank you.
Well, with that, Jason, drum roll, please.
Let's do it.
All right, Jason, what is your favorite dish to make?
Oh man, dish. See, the words matter, right? Like in marketing, like you said the word dish, so it implies that there's some kind of extra special time involved in recipes.
Oh no, man! If you say grilled cheese sandwiches are a dish, you just gotta,
It could be boiled eggs, and we'd have an opinion on it,
Yeah, of course.
Well, okay. Well, that makes me feel better then. So the tried and true ever since I was a kid has to be mac and cheese, right? It's easy.
That's great. It's not Kraft mac and cheese. Please tell me it's more than that.
Oh, it's any kind mac and cheese. It makes for a quick snack or a lunch, right? Particularly when you got various long hours or you're traveling or whatever.
Make a homemade version of this, or is it out of a box?
Both. Yeah.
Okay. Well, tell us about your homemade version, and do you add something cool to it? Because I've got like 6 or 8 different types of mac and cheese I like to make.
Well, now you're really
sometimes I'll put lobster in it. I've been known to put shrimp in it, and there's nothing better than a hot dog and a mac and cheese.
Yeah.
And then, what type of cheeses do you use? I like using a little bit of mixture of. I don't mind Velveeta, but I tend to stay away from it. My favorites are to use some Gruyere cheese and things like that.
Yeah, you got to mix it up. My wife and I made some mac and cheese together just a few months back, and we put we put too much cheese in it. and left the noodles on too long, and it was like this huge blob of like it almost looked like thick potato soup in some way. So you got to watch how much cheese and how long you cook the noodles. That's for sure.
Yeah, yeah.
But
And
Dom, yeah, you got to experiment with some different
you don't
cheeses.
put crumbles on it. You don't put anything on top of it. You sprinkle it with bacon. I mean, you're like oatmeal right now.
I got to tell you that boy, this is really tough. I didn't know this was going to be such an interesting,
Oh, come
topic.
on. And I'm sure I heard John cringe when he heard Velveeta. That's
Velveeta,
not
yeah.
his diet.
Yeah. Look, I'm not beneath saying that I will do the out of the box stuff. Like I don't mind it. I don't mind
John,
it.
you got any comments? Because we're going to move on.
I've got my hands over my ears. I'm going la 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 because I can't hear you.
That's so funny.
All right, you better have a better answer for this one, Jason.
Yes.
What's your
I'm ready.
favorite adult beverage?
Okay, well that's easy. That's the old fashioned.
Okay.
So
And
the-
do you have a preference of whiskey,
Well,
bourbon?
I usually like to if we go to a sort of a mid or upscale, I like to ask what they prefer, what their house is, or sometimes they have a unique blend. So I'll usually go with that. But I would say here's the sticking point, and I can dig in on this one is you got to have the big ice cube.
Ah,
Okay.
okay.
It can't right. It can't be like multiple.
I think, Jason, you and I are finding we've got increasingly less in common the more we talk.
But I think the difference is Jason would eat anything, I'm guessing, because I've hung around Jason a little bit. He's like a gremlin. If he doesn't get food, he's definitely like, "What are we eating?" And he's a skinny
Exactly.
guy, so it's amazing that because I'm always like, "Man, really?" And then he how about like, do you like a smoked one? I mean, or like when you Maker's Mark or Woodruff or anything that's your little Angel's Envy. These are all good because you don't want to take a great expensive whiskey and make it into a old fashioned. That's ruining some good stuff. But
Right. I'm not too terribly picky. I've personally, tried each of those myself. Kind of at home, different mixes. I do like a little bit smokier. I would say I'm not a fan of the odd taste. Like I went to a restaurant once recently, and it was like a peach something old fashioned. I'm like, I'll give it a shot, but it tasted like peach.
No,
and that's not what you want.
yeah, every time I order one that's a little bit different than an old fashioned, my wife's like, "You don't like that?" I'm like, "Yeah, and I'm just trying to grow." And every time I'm usually disappointed when I order something like that.
Same.
And you said you made them. Well, maybe for time, we're going to ask you because I do think I know we got to maybe wrap this up. What are your weekend plans?
Oh, that's good. Well, that we are in not only trade show conference season, we are in football season, American football season. So usually on the weekends, that's college football and pro football. So that's where we do most of our weekends, at least for the fall.
Oh yeah, I'll be heading to the KU game this weekend. That's part of my plans. It's KU K-State, which is a pretty tough ticket to get, but I am a season ticket holder, as maybe the audience has learned over the last couple months, and
Right.
that's a great. It's an 11 o'clock game, which my son's fraternity is right up the road from it, which is hence why we got the tickets because we get to see them. And we get to park there, and so we're going to have to get up at 8 a.m. But I have an even bigger event on Friday is the annual Halloween party that I attended. This is a big event for me. I mean, we planned for months with our costumes, and I think I've been going for about 7 years. There's usually about 50 people at this party.
Wow.
It's a big time party, and most of us are over 50 or some into our 60s, and then there's a spattering or maybe 30 to 40% of the crowd will be in their 20s, so it can get a little rowdy, if you know what I mean. But my wife and I are 3-time winners of there's a whole bunch of awards given out. We've won best costume 3 years. This year, we're going as Kiss, and this is before Ace Frehley had passed, so that's only somewhat timing. But us and another couple are going to get together sometime tomorrow afternoon and get my wife loves putting the makeup on. We got these great costumes. We got some guitar props. So it's a big weekend. I hope my old self can keep up with the youthful mind that I have because it's going to be a long weekend, but excited for all of it. The only downside is it's supposed to rain at the K-State football game. So we'll see how, I said K-State, the KU football game.
They're coming into town.
Slip. But John, what do you got
I'm
going
going
on?
to chime in quickly. So Saturday, Sunday, no idea what's happening, but Friday, Basically, yesterday was my Kath and my wedding anniversary. So Friday night we're going out for dinner because I had no idea when I was going to get home last night from Olathe, and as it turned out, I didn't get into the house till like 10 o'clock. So there wasn't much of an anniversary left to celebrate. But we're going to it was a restaurant in the village called Hazelnut Kitchen, which I've name checked multiple times on here, which sadly is closing. The chef wants to spend more time with his family, and so I was there about a month ago, and immediately I enjoyed my meal so much I booked like 2 or 3 more reservations, and one of them is next Friday to celebrate or Friday coming celebrate our wedding anniversary, and then we're actually there the last night before he closes up. I'm not sure what he's going to do with the space after that, but it'll be a sad loss to the village, and hopefully he'll continue to do the odd event here now and then. I just don't know what he's going to be doing, but anyway, we're really looking forward to it because I was there last week when Kath, my wife, was in New York, and I'd booked a table for 2, but as it was, I ended up being Billy No Mates eating by myself. But as the food was good, I didn't care. So yeah, that's it. I think Saturday, yeah, like I said, the rest of the weekend is fair game for what we decide to do.
And what about how many years have you been? You want to share how many years you've been married?
It's 37.
Okay, wow,
Congrats.
that is a big number. Good for you. I got 25 next year, but that's impressive.
Yeah, I think it's yes. I'm astonished it's 37, but it's 37.
I got a question for John. John, at this restaurant, do they have mac and cheese?
They do some really nice pastas because the dishes change a lot. But I talked about it a few months back. They did a dish as part of Green Crab Week because green crabs are an invasive species, and restaurants in various places are trying to figure out how to use green crabs to basically, help keep the population down. And they did this fantastic pasta with green crab sauce, and it was lovely.
I read about that, John. Did we talk about this on the podcast?
Yeah, we talked about it. I talk about it. Yes, that one pass. It links up so many things that are interesting me, like food, marine biology. Just
helping the environment, all of it.
Exactly. Yeah. So I think we probably should wrap things up. Jason, it's been an absolute pleasure to have you on. Maybe we'll do the same thing in about 12 months' time, unless we do it, in an intervening podcast. But yes, it's been a great pleasure to have you on. So thank you very much for joining us.
Yeah,
sounds good. It's been a lot of fun. Thank you, guys.
Thanks, Jason, and we'll see you in the funny page.